Welcome student PCH180151 Unit 4 Bioinorganic Chemistry. I am Dr. Devangar Pandya from Department of Chemistry, St. Xavier's College Autonomous, Ahmedabad. These are the syllabus points that we need to discuss in this unit. As far as the points are concerned, it's a very lengthy unit, but as far as information is concerned, it's very interesting and very important information we can obtain in this unit. We will begin this unit by the discussion of metalloporphyrin, that is enzymes, its definition. Then we need to discuss related hemoglobin and myoglobin, then cytochrome, vitamin B12, that is cyanocobalamin, zinc metalloenzymes, nitrogen fixation, essential and trace elements in biological system, biochemistry of non-metals like potassium-sodium pump, action of both ions in sodium-potassium pump that we need to discuss in this point, toxic metals and their toxicity, coordination compounds in medicine, chelation therapy, gold compounds and rheumatoid arthritis, anti-cancer drugs, platinum containing anti-cancer drugs, gold complexes, metallo synesthetic anti-malarial agents, metal complexes as radiodiagnostics agents, and magnetic resonance imaging. So very huge unit as far as the points are concerned, but as it gives very important and crucial information regarding the biological action in our human and animal body. So let's begin with our first point, that is metalloporphyrin enzymes and its definition. Porphyrins which are combined with a metal ion. Metal is bound equally to all four nitrogen atoms of pyrrole rings. They possess characteristic absorption spectra which can be utilized for identification or qualitative estimation of porphyrin and porphyrin bounded compounds. Metal porphyrins are important biological compounds, and as far as the example is concerned, iron containing hemoglobin molecules are metalloporphyrins that bind cooperatively to oxygen and act as oxygen transporters in vertebrates. Cytochrome C oxidase, an enzyme that contains iron porphyrin units, helps to reduce oxygen to water during aerobic respiration. Metalloporphyrins are also catalysts in other redox reactions involving hydrogen to hydrogen ion, which is an important for energy source, bacteria and algae, and nitrogen to ammonia, that is for nitrogen fixation, which provides nutrients to plants. Many have multiple metal centers that increase the rate of electron transfer in these reactions and reduce unwanted side reactions that form toxic products. Metalloporphyrin enzymes, this is the structure. What is porphyrin ring? Then porphyrin are tetrapyrrole macrocycles, 1, 2, 3 and 4 tetracycle macrocycles with conjugated double bond, one after the other double bond system is there, conjugated double bonds and various group attached to the perimeter, 1, 2, 3, 4, 5, 6, 7, 8 various groups attached to this perimeter. Variation of substituents facilitates the training of electron donating and electron withdrawing ability of the ligand. The porphyrins can accept two hydrogen ions to form +2 diacids or donate two protons to form -2 dianions. Porphyrins are found in many metalloenzyme like Fe porphyrin, that is going to found in cytochrome, and the function of that metalloporphyrins is electron transfer. Fe porphyrin is even found in hemoglobin and myoglobin, and the function is as dioxygen carrier. Magnesium porphyrin that is found in chlorophyll, and its main function is a photosynthesis. Hemoglobin, an elegant three-component system, has evolved to transport dioxygen from regions of high abundance water and add to regions of relatively low abundance and high demand the interior cell of the organisms. This process you can see in this figure. Now hemoglobin, the central component is dioxygen carrier protein. In the three chemically distinct carriers that have evolved and are found today, the dioxygen binding site in the protein, that is so-called active site, is a complex either of copper or of iron. For hemoglobins, the most widely distributed family of dioxygen carriers, the active site has long been known to consist of an iron porphyrin, that is heme group, embedded in the protein. Almost all hemoglobin share the basic structure illustrated in the figure. This is figure A, that is Fe2 protoporphyrin 9, that is also known as Hem B, hemoglobin and erythrocorins, is and the B chloroheme, that is chlorocorins, and the C is a myoglobin.
Each red blood cell contains 280 million hemoglobin molecules. Hemoglobin is folded protein with 4 iron-containing heme groups. Each heme unit binds with one oxygen unit. As a hemoglobin unit becomes oxygenated, its color changes to a brighter red. Collectively, this molecular color change is evident in the bright red color associated with arterial blood.
This is a myoglobin structure, in which you can see that there is porphyrin ring structure surrounded by the globulin part.
The binding of one oxygen molecule to the hemoglobin subunit changes the conformation of the subunit. This increases the oxygen affinity of the other subunits. The binding of oxygen molecules one after the other causes gradual changes in the conformation of the hemoglobin molecule. In tissues, the pH and the concentration of carbon dioxide in the environment change the structure of the hemoglobin molecule and therefore its affinity for oxygen. Protons, H+, and carbon dioxide can bind to hemoglobin, thus promoting the release of the bound oxygen. In lungs, at the same time, oxygen promotes the release of the bound protons and carbon dioxide. 2,3-biphosphoglycerate, BPG, which occurs naturally in erythrocytes at similar concentrations to hemoglobin, decreases the affinity of hemoglobin for oxygen. BPG passes through the canal in the center of the hemoglobin molecule and cross-links the beta subunits. This prevents conformation changes in the subunits. The resulting stable and unchanging conformation is unable to bind oxygen. BPG is therefore instrumental in regulating the amount of oxygen bound by hemoglobin.
Cytochromes are heme proteins containing heme groups and are primarily responsible for the generation of ATP via electron transport. They are found either as monomeric proteins or as subunits of bigger enzymatic complexes that catalyze redox reactions. History: cytochromes were initially described in 1884 by McMahon as respiratory pigments. In the 1920s, Keelan rediscovered these respiratory pigments and named them the cytochromes, or Euro pigmenta Euro, and classified these heme proteins, on the basis of the position of their lowest energy absorption band in the reduced state, as cytochromes A, B, and C. The UV visible spectroscopic signatures of HEMs are still used to identify heme type from the reduced bis pyridine ligated state, that is, the pyridine hemochrome method. Within each class, cytochrome A, B, or C, early cytochromes are numbered consecutively, for example CYT-C, CYT-C1, and CYT-C2, with more recent examples designated by their reduced state or band maximum, for example CYT-C559. Structure and function: the heme group is a highly conjugated ring system surrounding a metal ion, which readily interconverts between the oxidation states. For many cytochromes, the metal ion present is that of iron, which interconverts between Fe2+ and Fe3+ states or between Fe2+ and Fe3+ states. Cytochromes are, thus, capable of performing oxidation and reduction. Because the cytochromes are held within membranes in an organized way, the redox reactions are carried out in the proper sequence for maximum efficiency. In the process of oxidative phosphorylation, which is the principal energy-generating process undertaken by organisms, other membrane-bound and soluble complexes and cofactors are involved in the chain of redox reactions, with the additional net effect that protons are transported across the mitochondrial inner membrane. The resulting transmembrane proton gradient is used to generate ATP, which is the universal chemical energy currency of life. ATP is consumed to drive cellular processes that require energy. Types: several kinds of cytochrome exist and can be distinguished by spectroscopy, exact structure of the heme group, inhibitor sensitivity, and reduction potential. 3 types of cytochrome are distinguished by their prosthetic groups. The definition of cytochrome C is not defined in terms of the heme group. There is no cytochrome E, but there is a cytochrome F, which is often considered a type of cytochrome C. In mitochondria and chloroplasts, these cytochromes are often combined in electron transport and related metabolic pathways. A completely distinct family of cytochromes is known as the cytochrome P450 oxidases, so named for the characteristic Sirott peak formed by absorbance of light at wavelengths near 450 nanometers when the heme ion is reduced and complexed to carbon monoxide. These enzymes are primarily involved in steroidogenesis and detoxification. References: External links: Scripps Database of Metalloproteins, Cytochromes at the U.S. National Library of Medicine. Medical subject headings.
Cytochromes are electron transfer protein. There are 3 types of cytochromes depending on the porphyrin type: cytochrome A, cytochrome B, and cytochrome C. Only you can see the chain is different in the 3 structures. See, I have already explained you the structural difference in cytochrome A, B, and C. They differ with respect to cytochrome C with heme C, to substituents on the periphery of the heme group. Cytochrome functions as electron release, exploiting the switching of the ion centers from ferrous to ferric. In addition to cytochromes A, B, and C, many other cytochromes exist. These other cytochromes include cytochrome oxidase and cytochrome P450. History of cytochrome. Cytochromes were initially described in 1884 by McMahon as respiratory pigments, myohematine or histohematine. In the 1920s, Kellin rediscovered these respiratory pigments and named them the cytochromes. Cyto, that is cell-related, cellular, and chroma, that is color. So cytochrome, that is a cellular pigments, and classified as heme proteins on the basis of the position of their lowest energy absorption band in the reduced state as cytochromes A, cytochrome A absorb energy at 605 nanometer, cytochrome B nearly 565 nanometer, and cytochrome C at 550 nanometer. The ultraviolet or UV to visible spectroscopic signatures of heme are still used to identify heme type from the reduced by bispyridine liganted state. The pyridine homochrome method. Within each class, cytochrome A, B, or C, early cytochromes are numbered consequently: site C, site C1, site C2, with more recent example designated by their reduced state R-band maximum, that is site C559. Structure and function. The heme group is highly conjugated ring system which allows its electrons to be very mobile, surrounding a metal ion, which readily interconverts between the oxidation states. For many cytochromes, the metal ion present is that of iron, which interconverts between ferrous, that is reduced form, to ferric, that is oxidized state, electron transfer process. Cytochromes are thus capable of performing oxidation and reduction. Because the cytochromes as well as other complexes are held within membranes in an organized way, the redox reactions are carried out in the proper sequence from maximum efficiency. In the process of oxidative phosphorylation, which is the principal energy generating process undertaken by organisms, other membrane bound and soluble complexes and cofactors are involved in the chain of redox reactions, with the additional net effect that protons are transported across the mitochondrial inner membrane. The resulting transmembrane proton gradient, that is proton motive force, is used to generate ATP, adenosine triphosphate, the energy, which is the universal chemical energy currency of life. ATP is consumed to drive cellular processes that require energy, such as synthesis of macromolecules, active transport of molecule across the membrane, and assembly of Falcella. Many scientists believe that cytochrome C has evolved at a very fairly constant rate. The constant rate of change in cytochrome C may be the basis for the molecular clock. The molecular clock can be helpful. Here some videos are put to explain you about the function of cytochrome.
Electron transport chain or respiratory chain. It is present in the inner mitochondrial membrane. The electron transport chain consists of various electron transport or electron carrier molecules. The electron transport molecules are arranged in a sequence. They carry or transfer electrons from reduced coenzymes like NADH, FADH2 to the final electron acceptor, that's oxygen. The electron transport molecules are known as components of the respiratory chain. Some of them are proteins. However, non-protein carriers are also present. The position of a particular component in the respiratory chain depends on its redox potential. The components of the respiratory chain are arranged in the order of increasing redox potential. Starting compounds have negative redox potentials, and terminal components have positive redox potentials. Therefore, in the respiratory chain, electrons flow from negative to positive. In the respiratory chain, electrons flow from NAD to cytochromes via coenzyme Q and then from cytochromes to molecular oxygen. Coenzyme Q also collects electrons from FAD. The transfer of electrons from substrates to NAD and FAD is catalyzed by dehydrogenases. At the electronegative end of the respiratory chain, NAD-linked dehydrogenases, malate and glutamate dehydrogenases, catalyze transfer of electrons from substrates to NAD directly, but electrons from substrates like pyruvate and alpha-ketoglutarate are transferred via FAD. In contrast, some FAD-linked dehydrogenases, like succinate and glycerol 3-phosphate dehydrogenase, catalyze transfer of electrons from substrates to coenzyme Q of the respiratory chain because their redox potentials are more positive. Many components of the respiratory chain are present as complexes rather than single entities. On NAD, coenzyme Q and cytochrome C are present as individual components, whereas the rest of the components of the respiratory chain are present as complexes. The respiratory chain consists of 4 complexes and 3 mobile carriers. The complexes are complex 1, 2, 3, and 4, and mobile carriers are NAD, coenzyme Q, cytochrome C, and oxygen. All the complexes are integral membrane proteins. Each complex is involved in oxidation, accepts electrons from mobile electron carriers, and passes electrons to another mobile electron carrier. Complex 1. Complex 1 is NADH coenzyme Q reductase, or NADH dehydrogenase. FMN and iron-sulfur clusters are prosthetic groups of this enzyme. Electrons collected by NAD are transferred to coenzyme Q by NADH coenzyme Q reductase via FMN and iron-sulfur cluster. So NAD is oxidized. Complex 2. Complex 2 is succinate coenzyme Q reductase. It also contains FAD and iron-sulfur clusters. Complex 2 transfers electrons from succinate and glycerol 3-phosphate to coenzyme Q via FAD and iron-sulfur cluster. Complex 3. Complex 3 is coenzyme Q cytochrome C reductase, or cytochrome reductase. It also contains iron-sulfur cluster. Complex 3 transfers electrons from coenzyme Q to cytochrome C via cytochrome B, C, and iron-sulfur cluster. As a result of this, coenzyme Q gets oxidized. Complex 4 is cytochrome oxidase. It transfers electrons from cytochrome C to a final electron acceptor, oxygen. Hence, cytochrome C is oxidized.
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH. These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force. During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed. The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP. This enzyme allows protons to pass back into the cell and couples the energy released in this process to the phosphorylation of ADP to form ATP.
Respiratory complex 3 is a protein complex of the electron transport chain located in the inner mitochondrial membrane. Complex 3, cytochrome reductase, or coenzyme Q cytochrome oxidoreductase, transports electrons from the mobile electron carrier ubiquinone, Q, to another mobile carrier, cytochrome C. For each electron entering complex 3, approximately 2 protons are ultimately pumped across the inner mitochondrial membrane, and 1 cytochrome C, a mobile electron carrier, is reduced. Thus, for 2 electrons, approximately 4 protons are pumped into the intermembrane space. In fact, what is happening is that 1 electron is passed to an FES protein complex, then cytochrome C1, and finally cytochrome C, releasing 2 protons, while the other electron cycles through the 2 heme groups of cytochrome B, moving 2 protons from the matrix into the complex. They will pass through it, pumped by the next electron on the FES cytochrome C pathway.
This triumphant seedling is composed of many active cells. Inside each of these cells, countless chemical reactions are constantly occurring. To sustain these reactions, cells need abundant energy, which comes in the form of a molecule called ATP. Cells produce ATP during cellular respiration. Cellular respiration occurs in organelles called mitochondria. The mitochondrial electron transport chain is a key component in cellular respiration. The objective of this tutorial is to help you understand the steps of the electron transport chain. All eukaryotes have mitochondria. Let's focus on a plant. Each plant consists of several cells, each of which have mitochondria. A mitochondrion has an outer membrane and an inner membrane. Between the outer and inner membranes is a space called the intermembrane compartment. The inner membrane encloses fluid called the matrix. The reactions of the electron transport chain occur across the inner membrane of the mitochondrion. Cellular respiration is a multiple-step process. Glycolysis and Krebs cycle produce a small amount of ATP and also produce electron carriers called NADH and FADH2. The electron carriers then travel to the electron transport chain, where the electrons they deposit provide the energy to produce many ATP. In fact, most of the ATP that a cell generates is the product of reactions of the electron transport chain. The electron transport chain is a series of 4 proteins embedded in the inner mitochondrial membrane. So let's start our tour of the electron transport chain with the first membrane protein. NADH donates the 2 electrons it received during glycolysis or the Krebs cycle to the first enzyme of the electron transport chain. These 2 electrons will then travel through the second, third, and fourth proteins. Follow the arrow from the 2 electrons at the first enzyme through the other electron carriers. The electrons release energy as they travel through the proteins that make up the electron transport chain. As we will see, the mitochondrion uses this energy to produce ATP. Now, look at the second enzyme of the transport chain. FADH2, which is produced in the Krebs cycle, contributes electrons to the transport chain at this second enzyme. Like the electrons donated by NADH, the electrons donated by FADH2 also release energy as they travel through the third and fourth membrane proteins of the transport chain. Now, look at the fourth enzyme of the transport chain. Since this enzyme is at the end of the transport chain, it has to get rid of the electrons that it's acquired from the third protein in the chain. Otherwise, the whole chain will stop working as the carrier proteins become clogged with electrons. At the fourth enzyme, the electrons are getting dumped onto an oxygen, which is called a terminal electron acceptor. The oxygen then immediately reacts with free hydrogens to form water, one of the end products of respiration. Oxygen's role as a terminal electron acceptor explains why this gas is essential to the survival of most cells. So what happens with all the energy released by the electrons moving through the transport chain? Notice the hydrogen ions that are passing through the first, third and fourth enzymes. These enzymes use the energy released by the electrons to actively pump hydrogen ions from the matrix to the intermembrane compartment. This process creates a hydrogen ion gradient, also called a proton gradient, meaning that the concentration of hydrogen ions in the intermembrane compartment is now higher than it is in the matrix. Now, focus your attention on the ATP synthase protein at the bottom of the figure. If you follow the black arrow through ATP synthase, you'll see that the hydrogen ions enter ATP synthase from the intermembrane compartment and move through it to the matrix by facilitated diffusion. Since the hydrogen ions are moving from a high concentration to a low concentration, they're going from a high energy state to a low energy state and are therefore releasing energy. In a process called chemiosmotic phosphorylation, ATP synthase uses this energy from the hydrogen ion gradient to add a phosphate to ADP, producing ATP. The cell uses the ATP as an energy source that powers the cell's chemical reactions. For example, the cells of this seedling require much ATP to produce proteins and replicate DNA. Cellular respiration, and especially the electron transport chain, provides the ATP this seedling uses as it grows into a plant.
In the mitochondrion, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Tool in trying to determine when various organisms may have diverged from a common ancestor. The type of cytochromes. Several kinds of cytochromes exist and can be distinguished by spectroscopic structure of the heme group, inhibitor sensitivity, and reproduction potential. Three types of cytochromes are designated by their prosthetic groups. Cytochrome A has a prosthetic group heme A. Cytochrome B has a prosthetic group heme B, and cytochrome D that having prosthetic group as tetrapyrrolic chelate of iron. The definition of cytochrome C is not defined in terms of the heme group. There is no cytochrome E, but there is a cytochrome F, which is often considered a type of cytochrome C. In mitochondria and chloroplasts, these cytochromes are often combined in electron transport and related metabolic pathways. These are the cytochromes A, A3, B, C1, B6 and F, and these are their combinations: cytochrome C oxidase, that is complex 4 with electron delivered to complex by soluble cytochrome C, hence this name is AR3. Cytochrome Q, cytochrome C reductase, that is complex 3, and uh, plastoquinol, that is plastocyanin reductase. This way the cytochrome combines and they have different nomenclature, different type. A distinct family of cytochromes is the cytochrome P450 family, so named for the characteristic solid peak formed by absorbance of light at a wavelength near 450 nanometer when the heme ion is reduced with sodium dithionite and complexed to carbon monoxide. These enzymes are primarily involved in steroidogenesis and detoxification. Now, vitamin B12, that also known as cobalamin. Vitamin B12 is a water-soluble vitamin, key role in the normal functioning of brain and nervous system and for the homeostasis. It is a coenzyme for enzymes that transfer methyl groups and produce red blood cells. Vitamin B12 is composed of a complex tetrapyrrole ring structure, that is corine ring, cobalt ion in the center. This is the structure of vitamin B12, a very complex structure, and you can see this R that is attached to the axial side of the cobalt, and that R, if we are going to change, it will change the type of vitamin B12. If it is cyanide, then cyanocobalamin. If it is cyanocobalamin, that is known as vitamin B12. If it is hydroxide, then hydroxocobalamin. If it is methyl group, then it's methylcobalamin. If it is chloride, then chlorocobalamin. If it is adenosyl, then it is adenosyl adocobalamin coenzyme B12. So the substituent group differs differs the type of the vitamin B12. Here we need to discuss regarding cyanocobalamin. This is the planar structure of porphyrin ring. There's a cobalt in the center, and we have dimethyl benzimidazole attached in the axial side, and cyanide group remain attached on the upper side of that axial side, and we're going to get this structure, that is cyanocobalamin, vitamin B12. Derivatives of vitamin B12, cyanocobalamin, as already we explained, that is digested form. Hydroxycobalamin, if the R substituent group instead of cyanide group, there should be hydroxy group. Chlorocobalamin, instead of hydroxy or cyano, there should be chloride group. Then methylscopolamine, so instead of this cyanide, there should be methyl group. And adenosyl cobalamin, then that substituent group would be there. And 5-deoxy adenosyl cobalamin, that is another type of vitamin B12 derivative. Which source of vitamin B12? Natural source, that is produced by microorganism, bacteria or fungi, may inhibit in gut. Plants do not produce vitamin B12, and therefore we can have the vitamin B12 from the food source, which is rich in vitamin B12, that is liver, kidney or muscle, or egg, milk, cheese and other dairy products. Seafood and food fortified with vitamin B12 are also source of vitamin B12. How or where vitamin B12 stores? Normal body stores of vitamin B12 about 3 to 4 milligram primarily in liver. This would be sufficient for 3 years if dietary intake ceased or if the ability to absorb the vitamin was lost. Biochemical function. Vitamin B12 normally plays a significant role in one carbon transfer reaction, which in turn required for DNA synthesis, fatty acid synthesis, energy production. One carbon transfer reactions are required in biosynthesis of amino acids, serine, methionine, glycine, purine nucleotides, DTMP, that is deoxythymidine monophosphate. Enzymes that require B12 as cofactor are methyl malonyl, that is CoA mutase, isomerization of methyl malonyl CoA from odd C numbered fatty acids. Above reaction is an essential step for carbolism of fatty acids with an odd number of carbon atoms. Amino acids valine, isoleucine and threonine. When B12 is deficient, odd C numbered fatty acids accumulate in cell membrane of CNS, central nervous system, causing neurological symptoms. Methionine synthesis of methionine from homocysteine. Enzymes that require B12 as cofactor, that this kind of enzymes are there. Absorption of vitamin B12. Vitamin B12 absorption requires two binding proteins. Gastric acid and pepsin release the vitamin from protein binding in food and make it available to bind to cobalamin, a binding protein secreted in the saliva. Vitamin B12 is absorbed bound to intrinsic factor, a small glycoprotein secreted by the parental cell of the gastric mucosa. In the duodenum, cobalamin is hydrolyzed, releasing the vitamin for binding to intrinsic factor. Vitamin B12 deficiency. Liver can store up to 6 years worth of vitamin B12, hence deficiency are rare. Vitamin B12 deficiency develops as a result of a lack of intrinsic factor in the stomach, leading to malabsorption of vitamin. Vitamin B12 deficiency results in pernicious anemia, neurological complications, neuropsychiatric symptoms, gastrointestinal symptoms. Treatment for vitamin B deficiency. Folic acid vitamin B12 deficiency symptoms mimic, therefore folic acid and vitamin B12 both must be given to halt CNS symptoms. Well, this is enough from my end. Thank you for paying attention to this video lecture. Keep asking questions. As you know, asking question is a beautiful way to acquire knowledge. That's it. Thank you very much.